Alright, so let's say our boss is a really cool guy, and he gives us this auger plate. And on this auger plate there are some dots. Each dot represents a colony of duckweed, where in each colony there's one specific gene that we're looking at, and the gene is attached to a vector backbone, which I'll show you in a minute here. But one thing you'll notice is that there are green dots and that there are red dots. Well, the green dots contain living DNA, and the red dots contain dead cells. And that is because the auger plot has an antibiotic on it. Now, the reason why the green dots are able to stay alive is because they have the resistance to the antibiotic. And I'll show that to you right now. So essentially, this is what the green dot would look like at the cellular level. So you see, it has this insert, and it has this. We can use this square to represent the resistance to the antibiotic. So before we go into what we can do with that, let's talk a little bit about how our boss was able to make this auger plot. So essentially, what he did was he used restriction enzymes to cut out a piece of DNA, and the restriction enzymes are there to cut at a certain point, and so each different enzyme will cut at different points, and that can be determined by the nucleotide sequence. So let's say we have an enzyme that wants to cut anytime it sees CCC, GG, G, AA, TTC. So let's look at that right here. So we have A, T, G, C. So nothing there that we can cut because our enzyme doesn't want to cut that. Let's go a little further. C, T, A, no, nothing. And then we see this sequence, the C, 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 G, 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 A, A, T, T, C. And so that's where our enzyme wants to cut. And so let's look at this. Let's say we had a 1000 base pair sequence, and that's represented by this line here. So that this line has 1000 base pairs or nucleotides in it. And so let's say our enzyme goes along and looks for that sequence, the CCC, GGG, AA, TTC. And so it goes along, it doesn't find it, and let's say here it finds it. Okay, so that would be a cut. The enzyme would cut here, essentially just cut straight through that DNA sequence. And so it goes, the enzyme's looking, and it doesn't find it till here. It doesn't find that sequence of CCC, GGG, AA, TTC, and it can go through, and it's based on probability. So I mean, if you have an enzyme that maybe is engineered to cut at a place where there's even more nucleotides, it would probably be very rare that it would cut. So maybe in a 1000 base pair sequence, it would only cut one time. And so that all just kind of depends on how many sequences your enzyme is looking at, or how many nucleotides your enzyme is looking at. And so once we have these 4 different inserts, as you can see here, after the 3 cuts, you have 4 different separate sequences now. We can, or our boss, essentially, to make the auger plot, attached these inserts, that we can call them, onto a vector backbone, which is another sequence of DNA that's part of the duckweed colony. And also something to keep in mind too is that these inserts have the antibiotic resistance, which I'll show you here. So let's look back here again. So this green line, this green part of the cell, well, not really. Well, this green part of the plasmid would be the cut part, the part that was cut out from that 1000 base pair sequence, and this is the restriction, the resistance to the antibiotic. And this pink part here is the vector backbone, or the plasmid, and that's just what we connect our insert to. Alright, so now that we have looked at a little bit about how we've made this auger plot, let's kind of go into what we do once we have this auger plot. And so what we want to do before we do any experiments we want to amplify the gene. We want to copy the DNA that's in these living colonies. And so one thing to keep in mind here is that within each colony there's only one insert, which means there's one specific set of nucleotides that we got from our 1,000 base sequence. So let's say we want to clone our DNA, we want to amplify it, we want to copy it. So in lab we'd go and take a sterile stick, which I'll just kind of represent with a pencil, and I'll just take out the little colony and I'll put it in a test tube that has LB broth in it. And what LB broth is just a liquid that has nutrients that are essential for the bacteria to grow. So we go in and put our colony in there. So now that our colony is in there, we would just put this test tube with the LB broth and our DNA in an incubator, and that'll just allow it to grow, to amplify, to copy itself. And so after a day in the incubator, it'll multiply and grow exponentially. So once we do that, let's say it's the next day, and let's say we want to find out how many nucleotides are in our insert. So if you look back, we know that this insert was cut out from a sequence, but we don't know how long the sequence was or how long this is. We also don't know how many nucleotides are in this vector backbone. And so what we can do to find out the number of nucleotides is run a PCR reaction, or a polymerase chain reaction. And so to start that off, what we want to do is we want to what's the word I'm looking for? We want to dilute. There we go. That's the word I was looking for. We want to dilute this sample of our DNA. So we're going to add some distilled water, and then we're going to add some primer, some enzymes, which are primers. And what that does is allows these enzymes attach on to the vector backbone at specific points. And so if you look back here at this, we see that our primers latch on here at this black line and here at this black line. And so it attaches on into the vector backbone. And what's really cool is it's actually going to cut right here. And what we know from our primers is that it will cut at a point where there are 100 nucleotides between the insert and the cut on both sides. So we have 100 here and 100 here, plus the insert. So we have so after we cut this with our primers, we have 200 base pairs plus the insert. And so what we want to do after we do that is run gel electrophoresis. So let's kind of look at that here. So we have this gel, which is represented by the blue, and obviously this is at a microscopic level. So that's why our gels are kind of, you can see the individual parts, and these black lines are the DNA. And so one thing you'll notice is that there's a negative sign and a positive sign, and this represents the electrical charge. So DNA is actually negatively charged. So when we put our PCR sample into the wells, it will run to the positive side. And so one thing you'll notice is that the smaller DNA moves more quickly through the gel. And so the smaller, the farther it will move. The larger, it'll move less. So that's something to keep in mind. And so after we run this gel, we can actually look at and estimate how many nucleotides are in our insert. So let's look at that. Alright, so when we run our gels, we have to make sure we put in a marker, and that will tell us how many nucleotides are in each place. So let's say if our marker is here, there's 3,000 base pairs, and 2,000 base pairs here if our band was here. And so let's say our band from our PCR gels was right here, and this is around the 700 mark. Okay, and so the entire band that's included the insert plus the 100 nucleotides from each part of the vector backbone, that's 700. And so you minus 200 and you get 500. And that just is the amount of nucleotides in the insert.